So we'll start, we want to talk today, as I said, about soil building crops. And so this is some of the research that we've been doing through AIM and through Living Labs. And Judith and I, as I said, will talk first about some of that work that's happening through Living Labs and looking at different soil building crops and rotations that are aimed at improving soil health. And then we'll talk a little bit about potato early dying and the effect of rotation on that. So improving soil health. Lots of presentations and lots of discussion on this topic in the last number of years, but of course, what is involved with improving soil health? It's, we're looking at improving and cycling essential nutrients, providing essential nutrients through your organic matter and soil microbial community that's improving that. We're looking at the water holding capacity of soil organic matter. We're talking about soil microbial community and how they can feed and foster our plants and foster the microbial community itself to outcompete, say, bad fungi or bad bacteria that we don't want. How do we improve the aggregate stability and the soil structure to improve water holding capacity and improve the access to air in the soil? Because sometimes we don't think about that, but we need air for gas exchange in the soil as well. Protecting it from erosion, because often the soil we lose when erosion is the richest in organic matter. Outcompeting some of those soil-borne pests and diseases that we talk about. And overall, how do we have soil that's more resilient to buffer the effects of climate change? So we're seeing, particularly in our dryland system here in Prince Edward Island, we see more issues with periods of drought or periods of excess moisture or periods of high temperatures. So how do we have soil that's better able to buffer these effects? So how can we improve soil health? There's lots of different factors that go into improving soil health. Of course, there's reducing tillage and reducing soil disturbance. There's minimizing and mitigating soil compaction. Both of these top two bullet points are things that are a little tougher in potato rotation because we do have to do quite a bit of soil disturbance, but it's how do we minimize that disturbance? How do we reduce tillage? How do we prevent soil compaction in the first place? And when we do have soil compaction, how do we try and mitigate it the best way possible? We can improve soil health through using soil amendments like manure or compost. We're learning a lot more about how using cover crops to keep something growing, actively growing as much as possible is associated with improvements in soil health. Choosing rotation crops which break disease cycles and combat pests. We're seeing a lot more of that in the last few years, and particularly in PEI, as we look at things like battling wireworm or early dying through rotation. Choosing rotation crops with dense root systems that help to improve soil structure, help to build organic matter, and that's where perennial crops kind of come into play a lot. Leaving crop residues in the field where possible. So we are learning that building organic matter and soil health has a lot more to do with what's underneath the soil than what's above, but there is a value to leaving crop residue in the field where we can. And then all of this is predicated on preventing soil erosion and keeping soil in the field, because we need to keep soil in the field. We don't make soil very fast. It takes generations to build soil, and we need to keep it in the soil and keep that soil with the organic matter in it where it needs to be in your field. So these three are the in red are the ones we're mostly focused on today. We're going to talk a little bit about manure and compost as well, but we're primarily going to be talking about these options. We've talked about some of the other ones in some of our other presentations in the last year or so. So this is today's agenda. I've already gone over this a bit, talking about our full season soil building crops. Then we're mostly going to be talking about potato early dying and in a few different projects that we're working on. Give you an update on a mustard project we're doing with the Eastern Canadian Oilseeds Development Association. And at the end, I'm also going to give you a bit of an introduction to our Operation Pollinator project that we are working on. So leading off, Today, we're talking about comparing full season soil building crops planted the year before potatoes versus a check crop. And by a check crop, I mean what the grower would normally plant. So this is through Living Labs. Morgan and I have been working on this project with a bunch of different growers in the three areas, the East Prince Group, the Kensington North Watershed area, and the Surrey Watershed area. And these fields were first established in May and June and July of 2019, and in those crops, and then the potatoes were planted in 2020. And what we're looking at is we're testing for soil chemistry, soil organic matter, soil health metrics, verticillium and nematodes, soil nitrogen, soil compaction, and yield and quality in the resultant potato crop. Here are some of the treatment crops which we examined in the first iteration of this project. So hemp, buckwheat, sorghum Sudan grass, brown mustard, Sudan grass mixed with pearl millet, multi-species mixes. By this I mean 8, 10, 12 species in a mix, mustard and tillage radish, mustard and Sudan grass, and oilseed radish on its own. And in the first year, our check crops were a little more diverse. So we had red clover and timothy, red clover by itself, brown mustard, 3 fields with annual ryegrass as the check, and oats. As you'll see as we go forward, we've tried to maybe narrow down our list of check crops a little bit, but in the first year, this represents what the grower normally would have planted in those fields. What did we find? Just I guess a proviso before we get going. This is 1 year's data, so always kind of keep that in mind. We're not, not going to hang the hat on what we say today from these trials. It's just to start looking at some trends, start looking at some things we found, and that informs how we look at next year's trials or future year's trials. Last year was a challenging year in a lot of parts of the island. A lot of places were severely impacted by drought or by weeds or other things, early frost in some places. So that can have an effect on our data. So it's just something to keep in mind. So in 12 comparisons of alternative crops that were looked at versus check crops, the average difference between those two was about 1,500 weight an acre in total yield, and about 1,000 weight an acre in marketable yield, with a crop value of about $154 an acre. So this is as I said, this is just the trend over all these fields and all these comparisons. I'm not saying that this is statistically significant. This is just sort of looking at what we saw overall, the comparison. Some fields are very different than each other. We're looking at a diversity of crops and a diversity of check crops. However, there was 4 out of the 12 crop comparisons where the marketable yield was significantly increased where we had this alternative crop. And we did not have any where there was a significant decrease in marketable yield. So there was some where there was a numerical decrease that was slight, but it wasn't, statistically different than 0. So there was no fields where we really went behind, but some fields where we definitely came ahead. And we're going to go into a few more of those in a little more detail. We had 3 fields that did have a multi-species mix, and those multi-species mixes were reasonably similar. Across those 3, the average was that we saw no difference in yield or quality averaged over those 3 fields. One thing I would say is that in a couple of fields, there were some concerns about weeds being an issue, possibly weeds that were a little bit new to that field. And in one field that did have an increase in yield, we did see an increase over the check. The check was ryegrass, and that check was put in June, in July actually. And so it was a bit slow to establish, and it was a bit problematic to compare the two, because the biomass on the ryegrass was actually quite limited compared to what you would normally see. Just going to ask whoever's not muted to mute. I'm going to mute you anyway. Okay. Then on the sorghum Sudan grass, we had 3 fields that had sorghum Sudan grass. Again, the average of the 3 of those, that there was no difference in yield or quality. But in this case, we really didn't have any big negatives or positives. Like they were all very much not different from control for the most part. The one thing I would note is there was some other research last year where people were concerned about common scab following Sudan grass. We did not see any issues with scab. And in fact, in a couple fields where we did look at this, the scab was slightly less after the Sudan grass than after the control. So we didn't see anything to be concerned about on that. In 18 out of 22 treatments, root lesion nematode numbers increased. That is not a great surprise for us because root lesion nematodes have a wide host range, as we're going to talk about a little bit later. They generally increased the most after red clover and annual ryegrass, which were our check crops. So they increased in the checks more so than the treatments in a lot of cases. However, in most treatments, they increased by at least some amount. However, in 13 out of 22 treatments, the Verticillium dahliae populations decreased. So again, that's not entirely surprising because a lot of our treatments were grasses or grass-heavy mixes, and those are not known to be hosts for Verticillium dahliae. So we did see that Verticillium dahliae generally decreased the most following ryegrass, which makes sense. It seemed to increase the most after those diverse multi-species mixes. And again, that makes a certain amount of sense because in some of those 10 species mixes, there may be 6 or 7 different broadleaf species in them, clovers and brassicas and different flowering plants that all can be a host for Verticillium dahliae. I'm just going to pause for a second. Does anybody have any questions before I kind of go into some specific field examples? All right, I'll keep going. This was a trial that we did at Macaulay Farms. And this is what we have in the picture here. So on the left-hand side with the white flowers, this was Icarus radish, which is an oilseed radish. The middle was Timothy and red clover, and there's our check. And on the right was brown mustard, Centennial brown mustard. This was planted in early July 2019 to these crops. And then it was planted into russet Burbanks on May the 29th, 2020. This was in sort of the Dundas area, Mount Albion area of King County. And this was mowed and incorporated last year. Sorry, go back a little bit. The oilseed radish regrew a little bit and the mustard did not. So when we're looking at the soil testing that we did, there wasn't much difference in soil nutrients or in soil organic matter or soil health metrics between most of these treatments. We wouldn't expect to see much difference in the first year, but we did not see much difference. The nematode numbers did increase in all of the treatments. So I'm going to show you that down here. We need to put on mute. Okay. So the nematodes did increase. So you can see here, this is the before things were planted in the spring of 2019, and this is before things were planted in planted to potatoes in the spring of 2020. So you can see there was a difference to start off in the parts of the field. The radish, where the radish was going to be planted, already had almost 3 times the nematodes of the other 2 strips of the field. So you can see that all of the treatments increased a bit in nematodes the following year, but they increased more in the brown mustard compared to where they started than the radish, which only increased slightly. And then there was more than double the amount of Timothy, probably 3 times the amount in the Timothy clover, but it was still lower than the other 2 parts. These numbers of nematodes are moderate, I would say. They're not really high, they're not really low. On the verticillium side, again, you see there's a large difference between the starting points on the different strips, and these strips are very close to each other. So it tells you that verticillium and nematodes can change spatially in a field quite a lot in a small amount of distance. But on the verticillium side, it didn't change much in the mustard and the clover treatments. These are probably within, that amount of change is probably within the margin of error. So I don't think that they changed very much, but it does look like they decreased after the oilseed radish. So we'd be interested to see if we can replicate that in future trials in future years. If we can show that Icarus radish decreases verticillium, a lot of people would be pretty excited about that. So that's something we'll look at more going forward. Just going to move this to the top. This is looking at the yields. So everything that's in red is statistically significant at 90%, at a 90% confidence interval. So at 9 times out of 10, we expect that the reason that these are different is because of the change in the treatment. So for comparing the check with the oilseed radish, the only thing that was statistically significant was a bit of an increase in specific gravity. Everything else was not much different than 0. However, on the mustard side, we did see a significant difference, and we saw almost a 3,200 weight per acre increase in marketable yield and almost a $400 an acre crop yield improvement. And that's interesting because we actually, as I just showed, we saw that the verticillium and nematode numbers were higher on the mustard side than they were on the other two sides. So that's kind of interesting that they, even despite those numbers being a little higher, we still saw this yield improvement. So that gets us thinking that, you know, mustard may be having a beneficial effect on the microbial community. It may be having a beneficial effect on, it may be decreasing weeds. It might be making some nutrient more available. We're not 100% sure. But nothing that we measured last year could really pull out why that was different, except that we did see an increase in yield there. We also, this is a trial that we did with the Ramsays at Oyster Cove. This field was in winter wheat in 2018, and then was planted to fall rye as a cover crop. And then in early 2019, that fall rye was worked in, and we planted either annual ryegrass as the check or the Sudan grass pearl millet pea mix as the treatment. And then in addition, there was a fairly heavy dose of compost applied in September 2019 on both crop areas. So there was a place where there was compost put on, where there was ryegrass, compost put on where there was Sudan grass, and then places on both crops where there was no compost put on. You may be able to see in this picture here where I've got my cursor, there's a little bit of there's a line of manure here, and this is actually the compost turner that's beside it. So this was, you know, I believe beef manure that then was turned multiple times and made into more of a compost. And it was applied at 20 tons per acre, which is a pretty heavy dose of compost. But the Ramsays wanted to see whether by putting a big dose of compost on, we would see an effect. And then this was planted to Dakota russets in 2020. So in just looking at the effect of the compost on soil, so this is where we started, and this is comparing after compost, where we had ryegrass with no compost and where we had ryegrass with compost. And you can see there's a bit of a jump in organic matter here. Now, we would need to have additional samples probably over the field to verify that that's not just a change from sampling error, but it does appear that there is a trend towards a nice little jump in organic matter where the compost was applied. There was not a change in pH, but there was also an improvement in available P and K, and that makes sense with what was being applied. On all of the treatments, there was an increase in root lesion nematodes year over year. Those were, it wasn't really a trend that one side was higher than the other, and that compost didn't really seem to have much of an effect on that. The verticillium dahliae numbers were a bit more inconsistent, but they appear to have decreased a bit in the Sudan grass pearl millet pea treatment, which is this area right here in front of us. We did lose the Sudan grass pearl millet pea treatment with no compost. due to having to plant a different variety in that area. So they were a bit short of seed on Dakotas and needed to plant a different variety. So actually our check strip for that with no compost was over on this side of the field by the fence line, and that's where they had to put in a different variety. So we lost that, but we did have ryegrass with and without compost, and then the mix with compost. Our soil health metrics didn't change too much, although the active carbon and the respiration rate seemed to be a bit higher in all the treatments in 2020 versus 2019. Looking at yields, you can see there's on both of these, there's a significant impact on yield from the use of that compost. So you can see that compared to the check, we had almost a 4,400 weight improvement in marketable yield and a $550 improvement in crop value, where the compost was put on rye versus no compost. And then looking at that 3 species mixture versus the ryegrass with no compost, it's similar. It's about 5,000 weight with a similar crop value. So there's not much difference in these two crops that we looked at. Those values are fairly similar and we're not significantly different from each other, but they were very much different from our no compost check. So just summing up on that, the addition of compost had significantly higher yield and crop value than our check without compost. There wasn't much difference in the crop mixes, so the crops that we looked at. One large application of compost may have increased soil organic matter in 1 year, but that would require a bit more testing to confirm. The compost lowered the specific gravity by about 4 to 5 points, but in this, and that's consistent with other trials in other years, but we were still, you know, at max value on the contract for bonus, so that wasn't really an issue. And again, we saw no issue of common scab in any of these treatments, and particularly not with the Sudan grass pearl millet. Any questions before I keep moving? So Mary Kay had asked whether we're seeing a differing response from different crops in terms of verticillium and nematodes, and we very much are. And we're going to talk about that a little bit at the end too, about the effect of different crops on those. But it is, sometimes it isn't consistent year to year, sometimes it's not consistent even spring to fall. So we have to kind of take those results a little bit with a grain of salt and try and look at them over multiple years and multiple fields. But we definitely are associating certain crops with higher or lower effects on verticillium and nematodes. Any questions online? I better put the participant list up. I don't see anybody with hands up, so I guess we'll keep rolling. So we also then had a trial out in Victoria with the Vissers, and this was looking at mostly mustard mixes. So normally this field would have brown mustard as the normal crop for fighting wireworm, and they wanted to look at a couple of other mixes with mustard as well as a multi-species mix. So they had mustard and Sudan grass, which is right here. That's the mustard and Sudan grass together. They had mustard and tillage radish together, and then they had a multi-species mix with 8 different species in it, primarily with oats, peas, Sudan grass, and pearl millet. That would be the majority of the seed in the mix. These were all mowed once, and then they were planted to clearwater russet last May and irrigated. So again, we did not notice, there was no real difference in soil organic matter or most soil health metrics between the treatments, although we did see a bit higher active carbon respiration and biological end fixation under the mustard treatment. And that may indicate that under straight mustard treatment, that maybe that is invigorating or boosting the microbial community a bit. Not 100% sure about that by any means. This is 1 year and 1 year's trial, and these numbers were not massively different. So maybe it's just random information, but it was something we'll keep looking at in future years as we have mustard in treatments. The root lesion nematode numbers were inconsistent. They went up and down a little bit. They went down the most in the mustard, which is actually the opposite of what happened at Macaulay. So that's where we get into this inconsistency over root lesion nematodes. And then the starting values for Verticillium dahliae were very high across the whole field when we started. So some of the highest we had in any of the fields. That did not change much year over year. So there was no real difference between, before we planted these crops and after. So there was just small changes that are very much within the margin of error for that test. So then we looked at the yields, and again, I have these broken out, and Morgan, thank you to Morgan for all the statistical assistance with this. Where it's in red is where it is statistically significant at 90%. So we did see a significant difference in yield between the mustard and the mustard mixed with the tillage radish of about 3,300 weight and about $450 an acre. Some of that's due to improved 10 ounce, but wasn't statistically significant, but we do see a trend towards some better quality numbers here. On the mustard versus the mustard sorghum, it seems to be, there's maybe a trend towards some positive numbers, but it wasn't statistically significant, and we can't say it's different than zero, but maybe it's going in the right direction. We can say that we saw a higher 10 ounce and slightly fewer smalls, but not massive changes on the rest, enough to throw us into significance. This crop value is probably getting close, close to $300 an acre. It may be getting close to significance. And then on the mustard versus the multi-species mixes, these are not effectively different than zero. So they were pretty much the same. So again, we had significantly higher yield with the mustard radish mix than the mustard alone. The other treatments were not significantly different from the check, even though they were the lowest in yield and crop value in the multi-species mix. Verticillium didn't really change with any of the treatments, but the nematode numbers were a bit inconsistent, but they were not exceptionally high in this field. And there was a bit of an increase in the mustard Sudan grass treatment, but that was not significant, so we can't really report on that at the moment in terms of other than noting that it was numerically higher and maybe something to look at in another year. So where do we go from here in this project? We have 9 more fields that were set up in 2020 that will be in potatoes in 2021. So we have buckwheat versus red clover timothy mix. Again, we have a Sudan grass pearl millet radish mix versus ryegrass. Brown mustard or Sudan grass, they were planted side by side versus barley as a check. We have a couple with caliente mustard and radishes or multi-species mix. We have Sudan grass, oilseed radishes, alfalfa, another multi-species mix. So we've got quite a few crop comparisons to look at, and you can see that we've consolidated our list of check crops quite a bit, and most of them are with ryegrass. We have one with red clover and one with barley, but most of them are with ryegrass, so that these will be a bit more comparable year over year. And Morgan and I will be setting up 10 more fields this year in this project that then will be in potatoes in 2022. We're particularly interested in finding some growers that would like to compare, say, a traditional barley underseeded with a forage versus an annual crop like Sudan grass or mustard or radish. So we can kind of compare that low tillage but maybe higher nematode system versus like this annual cropping system and take a look at that. We're also had a few growers that are maybe interested in looking at corn ahead of potatoes. So versus ryegrass or clover, we might look at doing a field or two on that to see does that have any impact, negative or positive, on organic matter. As always, we're very open to suggestions and interests from growers in these areas that are part of Living Labs who would like to maybe be part of this trial. And outside of Living Labs, if there's growers in other parts of the province who are interested in doing on-farm trials related to rotation or tillage effects, we're always willing to hear those comments and talk to you about maybe ways that we could collaborate. So I'm going to pause. Does anybody have any questions about anything that I've presented so far? And before I turn it over to Judith. raise your hand or unmute yourself or put it in the chat. Okay, hearing nothing, I will turn it over to Judith.
Thank you, Ryan. So I'm continuing talking about the effect of manure and summer cover crop on subsequent potato crop. Okay. So this is a collaborative work with many AFC scientists. We collaborate on this project with PI Potato Board, and I want to acknowledge also the technical assistant from my team. So why do we want to test the role of manure? You know that potato bed system, they return minimal amount of residue to the ground. We also have increased soil compaction due to the heavy machinery and frequent traffic in potato. So maintaining and improving soil quality is challenging in potato systems. So manure can revitalize the soil with short-term yield benefit or long-term soil building benefits. We know that manure can increase water holding capacity, improve soil structure and soil buffering capacity, and cation exchange capacity. So applying manure on cover crop before potato can help to minimize the manure-associated soil-borne disease. Some growers, they complain to see increased scab when they apply manure close to the potato crop. So here we want to test what would happen if we apply manure 1 year before potato. So the cover crop, as Ryan mentioned, they are known to increase soil health. They can break the disease cycle. They can also build the soil organic matter. But we have to keep in mind that the cover crops are different. For example, if we consider the legumes, we know they can fix nitrogen from the atmosphere, and then they can decrease, our reliance to nitrogen fertilizer. But the timing of nitrogen release from legumes, especially in our conditions, if we do a fall plowing, we can have a synchrony between that nitrogen release from legumes and the time potato will need it. Grasses, on the other side, they don't fix nitrogen, but they have a good ability to mitigate soil erosion because they germinate quickly and they cover the ground quickly. They can also scavenge residual soil nitrate when you harvest a high demanding crop such as potato. So mixing grasses with legumes, we can find a benefit by mixing both. For example, superficial fine roots of grasses, they mitigate shallow soil compaction, whereas the tap-rooted legumes, they can reduce the deep soil compaction. They both can, if they mix together, increase the nutrient recycling because they have difference in root architecture and the growth pattern is different, so they can recycle the soil nutrient from deep layers. As you know, when you have a legume that fixes nitrogen, the fixation from the atmosphere is going to be inhibited if you have high available nitrogen in the soil. When you mix grasses with legumes, you can actually have that continuous capacity of legumes to fix nitrogen because the grasses will uptake the nitrogen released. So the other advantage is that the decay rate when you mix legumes with grasses is going to be modified. So the carbon to nitrogen ratio is going to be modified. So you can have a better synchrony between your nitrogen release from a mixture of legume and grasses and the crop nitrogen requirement, and you can minimize the nitrogen leaching. So the objective for this is to assess a broad range of cover crops where we have grasses, legumes, and a mixture of legume with grasses, with and without pen-pack cow manure. So we applied 20 tons per hectare of fresh manure, and we have a large field, it's 2 acres, where we have 4 replicates of each treatment. So here I'm going to walk you through the different cover crop we tested. In this column, you are seeing the seeding rate of the cover crop. And on the right side, you have the amount of fertility applied. So the rule is simple. When you have a grass, we applied 400 pounds per acre of triple 17. And when we have legume in the mixture, we applied half of this fertilizer. So we had alfalfa mixed with orchard grass. We have sorghum Sudan grass. We have red clover. We have rye grass, common veg, and crimson clover mixed together. We have forage per millet. We had sorghum Sudan grass mixed with Verticillium resistant alfalfa. We have winter rye mixed with hairy vetch and forage sorghum grown for 10 weeks and then followed by brown mustard. So here I am including the forage sorghum and the sorghum Sudan grass, just because I want to see if there is any difference. As you know, the sorghum Sudan grass is a hybrid between forage sorghum and the Sudan grass. Sudan grass is more shorter, the stem is more thin, and the forage sorghum on the other side is more taller, it's more leafier. So the hybrid is in between these two crops. So these plots were tilled and rolled before planting cover crop and rolled again after planting. The manure was applied 5 weeks before planting cover crop, and where we had manure, no fertilizer was applied during cover crop phase. So the seeding was done on June 8th, and for all crops, we had 2 cuts for all cover crops, except for red clover, where we had 3 cuts in that growing season. So when we were cutting the cover crops, they were flayed mold, and the biomass was left on the ground. So the first cut happened 6 weeks after seeding to control weed, and the second cut happened on October 2nd. All plots were floor proud, so everything was done in fall. So this is a picture that is showing you in the late summer how those crops looked like. So 2017 cover crop and 2018 potato. So what did we analyze? So we did take the biomass of the cover crop at every single cut, then we add it all together for the whole growing season. We analyzed also the total carbon and nitrogen. We did analyze the soil nitrate in fall following cover crop incorporation, and soil nitrate during potato growth by doing monthly soil sampling at 2 days. We did check the potato yield and soil properties, and we analyzed something called the soil nitrogen supply using a plant bioassay approach. So this is done by analyzing total nitrogen accumulation in potato, in vine and tuber, under the plot without nitrogen fertilizer application. So this is done before vine senescence, when you still have your whole crop. So we do the potato and the vine, and we combine the nitrogen accumulated. And this gives you an idea how much your soil can supply in terms of nitrogen. And it's a good test because it does integrate your growing season conditions, so your moisture and your temperature, it does reflect your growing season. So it's really a good index of the ability of your soil to supply nitrogen. So here I'm showing the cover crop dry matter biomass. So on the vertical axis, we have the biomass in tons per hectare. So when you have different letters, that means you have statistical differences. So what we did see is that the forage per millet has higher dry matter biomass. So the red clover is highlighted in red. So when you have the same letters, that means they are comparable. So we had comparable biomasses for red clover, ryegrass, common red, and Kim's clover, and sorghum Sudan. So what we would expect is that the carbon returned to the soil is proportionally to your biomass. So that's why in the next slide, we are seeing the total carbon accumulated in cover crop, and this is reflecting the biomass that is incorporated. So higher carbon accumulation with forage per millet. As you would expect, the nitrogen accumulated was higher with red clover, statistically higher than any other tested cover crop. And this is reflected in the amount of nitrogen, nitrate, soil nitrate involved. So we did see that the red clover was associated with higher nitrate in the top layer, so 0 to 6 inches or 0 to 15. And this was also reflected in the bottom layer, so 6 to 12 inches or 15 to 30 centimeters. So red clover was associated with higher accumulated nitrogen, and that was reflected in the amount of soil nitrate. And alfalfa mixed with grass was associated with lower nitrate in the soil. So when we did that estimation of soil nitrogen supply by quantifying the total nitrogen accumulated in vine and tubers in the plot without fertilizer, we did see a significant increase of nitrogen supply where we had manure. So no manure is in blue, and with manure is in orange. So you can see that manure increased soil nitrogen supply capacity. So without manure, we had values ranging from 38 to 60 kilogram nitrogen per hectare. And when we added manure, we had values ranging from 54 to 81 kilogram nitrogen per hectare. So this can be credited from your soil if you have to apply fertilizer. So let's see here the total potato yield. So we did see significant effect of manure and cover crops with higher total yield with forage per millet and forage sorghum, but the values were comparable with alfalfa mixed with orchard grass, sorghum Sudan grass, and winter real and heritage. When we included the effect of manure in total yield, we did see a significant increase of potato yield, total potato yield with manure. The increases ranged from 19 to 35 yield increases. For the marketable yield, we did observe again that forage per millet, forage sorghum, sorghum Sudan grass, and alfalfa mixed with orchard grass were associated with higher marketable yield. We analyzed the soil organic matter after harvesting potatoes, so 1 year later. Even if we did not expect to see a big jump in soil organic matter levels after just one growing season, we did see that there was a trend, a slight trend of increased soil organic matter with manure application. So we analyzed the soil respiration that is affecting the metabolic activity of soil microbial biomass. And we did see significant increases of carbon dioxide after incubation with manure application. So to summarize, manure increased potato yield and nitrogen supply capacity, and they were trained toward increased CO2 soil organic matter with manure application. The pearl millet and sorghum Sudan grass were associated with higher marketable yield and lower soil nitrate than red clover. And we did see higher total nitrogen accumulating in red clover and higher soil nitrate in fall, but this was not translated in increased yield. Total nitrogen accumulation in red clover and soil nitrate was higher than treatment where grasses and legumes were mixed. And cover crop returning high residues and manure are effective means to boost potato yield. So what is next in this project? So this project is still continuing, so we are starting the second phase. So in 2019, we did establish the same treatment, and the manure was applied in 2019. But instead of having just one growing season of cover crop, we are going to have 2 growing seasons. And in 2021, this summer, we are going to have potatoes in all crops. So to increase the ground cover in those plots that were going to forage pearl millet, forage sorghum, and sorghum Sudan grass, we did underseed barley with ryegrass. So to make this plot comparable to have a certain ground cover during the fall of 2019. And this growing season, all plots where we have a 2-year forage, so the plots in black, they were plowed in fall, whereas the plots where we had forage pearl millet, forage sorghum, and sorghum Sudan grass, we did not plow them. So they will be plowed in spring before seeding potatoes. So this treatment here, we have equal amount of soil disturbance. So we are going to do additional analysis. So soil microbial diversity will be added, and the phytopathologist will be assessing the scab and pyridine complex. And we have the weed scientist who has been monitoring the weed pressure from the year 1 up to the potato phase, and an agroeconomist who will be assessing the net revenue. So this is it. If you have questions, I will be happy to answer them. Thank you.
Does anybody have any questions for Judith? Oh, you want to unmute yourself and put it in the chat? Any questions? Well, I lunch today. I know some of the questions or some of the discussion we had at the local meetings yesterday and today was just looking at the trend that you saw, Judith, in that it appears that in the first cycle, you had the best marketable yields with after the C4 graph. So that was kind of interesting. We'll see whether or not we replicate that again this rotation. I think maybe there is something up in the chat. I'll come over and I'll look at that. Chat. Yep, we have a couple in the chat. So Barry asked, would soil respiration be a good indicator of organic matter change?
I think it depends on what you are looking at. There are some what we call the air indicators of, soil organic matter. So that can be an air indicator. Sometimes the total organic matter is not going really to show, to be shown in very short term, whereas the soil respiration can show, because there are different way to see the effect of organic, the soil building crops. So they can increase that stable carbon, but they can also act on stimulating the soil microbial activity, which is good because it can contribute to increase yield either by promoting the beneficial soil organism. So it's a good indicator because I did not show the active carbon, but the soil respiration here was a good indicator than the active carbon, where we did see very, very comparable values, even where manure was applied. So it's just checking more than one indicator of soil organic matter.
And then there's a question from David about the potato variety that was used.
It was Russet Burbank.
Right. Any other questions? Okay, we can, if you're thinking about some, we can have them at the end too.
Okay.
All right. Get my hand sanitizer going again. All right. So that brings us back to the question that I get a lot, which is, what should I be planting? What should I be putting into my rotation? And of course, that is a multi-layered question. And for me, it always depends on what is your biggest issue. You got to tackle the biggest issue first and try not to make the other issues worse. So if your biggest issue, if the thing that is, you're a table stock grower, or you're really battling with wireworm, and that's representing the biggest potential loss of sales on your farm, then that's the thing that you need to tackle first. And so that's what you'd be looking at, your brown mustard or your buckwheat or something to help try and reduce those populations and reduce the damage. If wireworm, maybe you're in an area that doesn't have so much wireworm, or you're not as concerned about that, you can manage it with insecticides or other cultural management, and you're really worried about early dying, well, then that's maybe where we start looking at the pearl millet or biofumigant mustard or Sudan grass, one of these types of crops which has been related to decreasing the impact of early dying in other trials. Maybe you're not as worried about early dying, you're planting a resistant variety, and you really want to get your organic matter up. You want to improve the water holding capacity of your soil. Then that's where we look more at, your perennial grasses and legume mixes, and how those could, and reducing tillage and having them be out for more years, and maybe that can help to improve your organic matter. And then maybe you're really worried about soil compaction, and that's an issue within parts of a field, or a field that you've got maybe on a little tighter rotation. That's when you're going to be looking at things like alfalfa with its deep taproot that can help break through compaction layers. Again, your Sudan grass and pearl millet are associated with helping to fight compaction as well in certain areas. The thing I talked about with some of the growers yesterday and today was, if you've got a field that has a history of strong yields and no early dying, and you've tested it, and your soil tests are really good and you're really happy, and it's over 3% organic matter, and you say this is a really strong field, maybe that's the field where you consider maximizing your cash crops. So that's maybe where you say, I can get a cash crop off that field every year. Maybe I can go barley or wheat after my potatoes, and then I could go peas the next year or something like that. Or, there's a few different options there. I can grow mustard for a cash crop. There's a few different ways you can think about that. There is an opportunity for continuing to maintain your soil health by putting in cover crops after those crops, so you can, I'm not saying you want to run down soil either, but not every field is under the same rotation, under the same dynamics, and there's a way to, possibly just look at what ways can you balance the need to maintain and improve soil while also making money. Right. So Mary Kay mentioned tillage radish. My one concern over tillage radish when it comes to, say, compaction fighting is that mostly I've seen that when tillage radish goes down and hits that compaction layer, it then pops out of the ground. So it can't actually break through hard compaction layers. It does do a good job of breaking up surface compaction, and it helps with water infiltration, and it can also scavenge nitrates and make those available the next year, especially if it's mixed with a grass. So if you mix it with oats or you mix it with rye or something like that, it could do some good things. But if you're planning in PEI, if you're planning on using tillage radish to just break compaction layers, it's not likely going to happen. So along this line, I have some additional resources to point you to. We have on our agronomy website, which is the address here, we have some of these other resources. So last year there was a presentation on choosing the right rotation crop that goes a little more in depth on each of these individual crops and, you know, pros and cons and that sort of thing. So feel free to check that out. I also did up a rotation crops quick reference table that has sort of, it's just a quick, you know, things to think about and things to look at, seeding rates and fertility needs and what they're good for, pros and cons. And that's just like one page front and back. So you can, and I just updated that in December. And then there's also a presentation from a couple more years ago that's bigger on tools for soil building, so that we go a little bit more into tillage and residue tillage and compost and manure application and a few different things. So those are all on the agronomy site, along with lots of other resources from other places, as well as other research we've done here in PEI. Then there's this Eastern Canadian cover crop tool, and I'm a big fan of this tool. That's the address for it here. And this is great if you're trying to, you know, you're looking at your rotation and you're looking at, say you're looking at when you're going to put cover crops on, or you're looking at when you're going to establish a forage mix or something. It has a whole big list of different crops and mixtures. This is only just part of them. It's kind of cut off because it's just a screenshot, but there's all these different mixes and crops. And then on the top, there is a list of, there's a date range, and this gives you the establishment date range for these different crops. Yellow is sort of iffy or you know, sometimes. Green is reliable establishment date windows, whereas blue is for frost seeding. And then each of these individual crops, you can click on it and it'll also show you a fact sheet on that crop as well. So, and this is specifically geared towards each individual province. So when you go to it at the top, I don't have this up here, you go to the top, it allows you to select Prince Edward Island. So, and it gives you the dates specifically for PEI or specifically for the province you're interested in. And actually it's different depending on county. So if you select Kings County as opposed to Prince County, you'll get a little different change in your dates here. So this is a really good little tool, and I use it quite a bit for extension purposes and for looking at cover crop establishment dates. So it's a good thing to try. I know here, like the winter cereal rye, here it'll say reliable establishment to about October 15th, and then iffy up to about, you know, the 25th, something like that. So. And then a shout out for the soil health test. So note, I had this question yesterday. This isn't a sample from our elite seed farm. That's the account that I built it under. This is from one of the, so our elite seed farm wasn't at 2.2% organic matter, but this was just a trial field. I erased what farm it was from. But this just shows you the soil health test that's available from the department. In it, you get a soil texture analysis, you get these different soil health characteristics, you get the actual test result, a score out of 100, and how that is rated compared to other island farms. And the province also has a handy page on how to interpret your results. So feel free to go and check that out too. These links will be available you know, if you want to access this slide deck later on, so you can look at that as well. All right, I'm going to pause for a second before I get into the early dying section. Does anybody have any additional questions on anything I just presented or anything on those sort of soil building crop discussion? Okay, if not, we'll go into the early dying section, which is the last main section of the presentation. So just a little refresher on potato early dying for those that are not as familiar. Most of you will be. Potato early dying is largely a complex of Verticillium dahliae, which is a fungus that lives in the soil, and root lesion nematodes, which is, of course, they're microscopic worms that live in the soil. And we're particularly interested in the species called Pratilancus penetrans. Verticillium dahliae, the microsclerotia live in the soil or are in the soil, and they enter the plant through the roots and essentially clog up the water transportation tissues of the plant. So the xylem and the phloem, and they block the plumbing, and they make it hard for the plant to uptake water. And that's why we get symptoms like this, where this is very traditional early dying symptoms, where we get the leaves are starting to wilt, and there's a discoloration in the rest of the plant. If we were able to go closer, you'd probably see there was some darkening of the stem, and this, compared to some of the other plants, this one is dying early. Where this makes it an additional issue is where we have root lesion nematodes, because the nematodes, they feed on the plant roots. They're a plant pathogenic species of nematode. And they, by nibbling and gnawing and entering the roots of the plants, they create entry points for verticillium. So they they exasperate the problem. They make it worse, and they increase routes of entry for verticillium. So it's not 1 + 1 = 2. It's often 1 + 1 = 4. It's really makes things a lot worse. Some recent research out of the United Kingdom also seems to indicate that root lesion nematodes and free-living nematodes may also have an impact on increasing blackleg symptoms. So the nematodes could be eating on a potato plant that where the seed piece was infected with blackleg bacterium, and then they go and gnaw on another root of the neighboring plant, and they bring that bacteria with them. So this is very new research, and it's a kind of a new thing, but they have seen it in a couple of different trials now. So they're starting to think that that might be a way that blackleg can spread likely with high levels of blackleg in seed and with high levels of nematodes in the soil. Having verticillium alone can cause early dying on its own, but the presence of nematodes makes it much worse. So this is what Verticillium dahliae microsclerotia look like, the little tiny peppery black dots. You can see all this vert clogging up the stem of the plant. And then this is a root lesion nematode. And you can see they have this little needle-like mouth part at their mouth, and that's what they use to pierce into the root, and that's what they use to nibble and make an entry point into the root. So most broadleaf plant species can be a host for verticillium. Preferred hosts are things like potato, of course. Field mint is a big one. Peppers, tomatoes can be as well. Soybeans and forage legumes seem to also be a preferred host. We think that, or we see a lot of evidence that grass species generally do not host verticillium. Almost all crop species can be a host for root lesion nematodes, on the other hand. So there's very few plant species that don't in some way host root lesion nematodes, but some are worse than others by a long shot. So the preferred hosts, again, potatoes are bad, corn, red clover from our research seems to be quite bad, and we're going to show you a bit more about that. Soybeans again, and annual ryegrass also appears to be a bit of a multiplier of nematodes. One that we know that is not a host is forage pearl millet, and that's one of the main reasons it's being grown, is it doesn't seem to host nematodes, and therefore nematodes generally go down in a field after pearl millet, because unlike verticillium can last in the soil, those spores can last in the soil for 10 years or more. And so they will gradually decline over time if there isn't a host species or preferred host species present. Nematodes, if there's no host there, they'll die much more rapidly. They don't have spores like verticillium. They're live creatures, and so they don't have a host to multiply in. They will gradually decline. Plus, nematodes, we lose a certain amount of nematodes in the soil every winter when we get frost. So over time, if you don't plant host crops for nematodes, they'll go down. The only problem is almost everything is a host for nematodes. So that's where we start looking at our not as bad hosts. So things like grain crops, like barley and oats, tend to not be as much of a host for nematodes as, you know, potatoes and corn and other things. Another plant species that isn't a host are marigolds, marigold flowers. And they've been planted, I know, in you know, plots in some cases to bring those down. It's pretty expensive seed, so it's not really feasible on a field scale, but they are also not a host species. This is just a slide I borrowed from Mario Tenuta. The general gist of this slide is that where they looked at the amount of early dying disease incidents, generally where they had low levels of disease incidence was also where they had the highest organic matter. And where they had the most disease incidence was where they had the lowest organic matter. So there is an effect that, you know, just because your verticillium numbers are high, just because from the level of pathogens that you think you're going to have a lot of early dying, if you had a very high organic matter soil, that may help to buffer some of that. So some of that may be some of the other critters in the soil help to outcompete the verticillium and the nematodes, and maybe even eat some of it. But also it could be that those high organic matter soils hold more moisture and are more resilient against moisture stress, which is also exacerbates early dying. So the last couple of years, we've done a survey as part of this CanPEDNet project under the CHC cluster. So CanPEDNet, Canadian Potato Early Dying Network. And myself and Sebastian with the Department of Agriculture, and Morgan has been a big help on this. We've been doing surveys of these fields in cross PEI. We do 30 fields each October, and we're measuring verticillium and nematode populations in those fields. And there are a variety of fields in a variety of rotations coming out of a whole variety of rotation crops. And the only thing they have in common is that they're supposed to have russet Burbank potatoes in them next year. So these are the fields we did in fall 2019. You can see the nematodes, they had a low of about 1,800, which is reasonably low. A high of almost 28,000, which is very, very high, and an average of 10,000. 5,000 nematodes per kilogram of soil is the previously agreed upon threshold level for damage in russet Burbanks. So 19 out of our 30 fields were at or above that threshold, and the average was twice the threshold. So that tells us we have lots of root lesion nematodes on average in a lot of our fields. The same way, the verticillium, this is done using PCR, and we're measuring cells per gram of soil. And in this case, we had a low of 234, which is actually quite low. And this was in a field that hadn't had potatoes in it in 20 years. And then a high of over 13,500. And this would be probably one of the tighter rotation 2 or 2.5 crop rotation fields, and an average of over 7,000. And we don't really have a threshold right now for from this test as to what the verticillium is that causes damage. That's one of the main reasons we're doing this work. But generally, anything over 3,000 that I've seen seems to be reasonably high. And we had 26 out of our 30 fields that were over 3,000. So again, the only fields that didn't have a lot of verticillium were the ones that hadn't had potatoes in a really long time. And generally, the only thing that really decreases verticillium over time, as I said, is distant time between potato crops. So this, again, this puts it a little bit in perspective of splitting it up by the preceding crop that was in these fields. So you can see the blue is the nematodes and the orange is the vert. And you see the nematodes are much more variable depending on what they were coming after. So where there was a hay crop, it was some of the highest. Also with the soybean and peas was very high. But there where we had grain that wasn't underseeded, like barley or wheat, it was actually quite low. Like it was only about 2,000, which we would say is, you know, below threshold for most varieties. Pearl millet was also fairly low, and that makes sense. This was only one field of radish that was a bit low. That's, as I said, it's only one field. The rest of these were all multiple fields. And the grass and legume, this is like 15 fields. So the average over those is pretty high. On the verticillium, you can see they don't vary quite as much. They're all high, and they're just high by varying degrees. But the lowest is this one field of radish, which is still over 4,000, which is still a lot. So moral of the story is verticillium is high in most of our fields if you're in a 3-year rotation or 4-year rotation, and it's harder to manage. It's easier to manage nematodes. I'm not going to say it's easy, but it is something that we can manage. So then we took those 30 fields and we identified them as, high, medium, low inoculum fields. And we identified what we thought were 4 of the highest inoculum fields and 4 of the lowest inoculum fields. And then we went back in and followed them through the crop season. So we went in and we took 5 GPS points in the field. We did follow-up soil sampling there. We did, and then we measured the amount of amount of verticillium wilt and early dying in the foliage at multiple times during the year. Morgan and Sebastian did a lot of that this summer, and we were rating the leaves and rating the plants on a scale as to how much damage there was. That data has been collected and is being calculated right now. I don't have that to share with you right today. And then we did yield and quality at those same 5 sites again in the fall. So as I said, these are the high inoculum fields. So on average, these are the ones that were high for nematodes and verticillium. You can see we have a variety of yields here. 3 of these were in Kings County, which had more rain. Actually, a couple of these are probably better than we would have expected for how many nematodes and how much verticillium they had, but I think they were made out a little bit better because they did have some rain. This one, Kings 3, had Dakota russets. It was the only field of these that had Dakota russets, just because of, at the last minute, Dakota russets went in there instead of Burbanks, but the rest of them are all Burbanks. This is the lowest number that I have gotten in a field trial in my career with the potato board so far, which is less than 70 hundredweight. So that's very, very low. You can see it was on a short rotation length, and it had very low organic matter, which was exacerbated these numbers. You'd say actually the nematodes weren't that high comparatively to some of the others, but the verticillium was high and the organic matter was really low. Now in comparison, these were our low inoculum fields. So they started out a bit lower. You can see the verticillium generally, a couple of them are quite low, and those are in our long rotation fields. But a couple of these long rotation fields were very affected by dry conditions. So they were non-irrigated, had a very low amount of rain, and so did not do great on yield either. But we did have a couple here in the 300s for sure. So if we compare these, our high inoculum fields and our low inoculum fields, there's 80 hundredweight difference between them, just an averaging the 4 of each. And the high inoculum fields had twice as many root lesion nematodes and 3 times as much verticillium as the low inoculum fields. And this is despite, as I said, these high inoculum fields, 3 of them were in Kings County where we got some rain, whereas only 1 of the low inoculum fields was in Kings County. So just something to consider. So again, this is 1 year, this is 8 fields. The purpose of this survey and this CampEdNet work that we're doing with the board is really trying to identify what level of verticillium and nematodes is associated with what level of yield loss and what level of risk of yield loss, so that growers can make a decision. Okay, based on my test results, do I have to look at using Bellum Prime or Aprovia or one of the types of in-furrow treatments? Am I going to use a resistant variety instead of Russet Burbank or another susceptible variety? Am I going to plant mustard or pearl millet or something in that field to help me try and bring those numbers down? So it's about providing you with more options and more opportunities to make changes. There are other parts of the CampEdNet project that are looking at other things. So there is some work going on looking at, again, the effect of these different products. They're looking at the effect of rotation crops, biofumigation, Whether we can see different strains of verticillium, different strains of nematodes having maybe different effects. So yeah, there's a lot to this project. Then we're doing an early dying project through AIM, where we're looking at the effect of comparative effect of different legumes. So we're trying a lot of questions from growers on, you know, is red clover that much different than other legumes or other forage species in terms of how it multiplies verticillium and nematodes? Do I have other options? If I want to keep forage in my rotation, but I don't want to make nematodes worse, what are my options? So that's why we started this trial in 2019. So we have 2 fields in West Prince that were 2 years of forage, and they're going to be potatoes this year. And they had these treatments. So we had double cut red clover, alfalfa timothy, white clover and festolium grass, bird's foot trefoil and festolium grass, and then festolium by itself, or Italian ryegrass. We had 1 field where the festolium by itself didn't do that well, so we compared it with Italian ryegrass instead. I will also say that the bird's foot trefoil didn't establish super well either. 1 field it didn't establish hardly at all, and the other field it really only came on in the 2nd year. So we omitted this from our stats and from our presentation just because I don't think it's really a fair representation of this treatment. So we're going to look at the other things. This is what the trial looks like. So this is alfalfa timothy. This is double cut red clover. Over here, I think the next one over was bird's foot trefoil, and then it was white clover, and then it was the grass over on the side. So this is looking at our verticillium. So we started with a composite sample of the field, the first field back in May of 2019. It was very high for verticillium. You can see then in October of that year, not a lot of change, a little bit of, not much change in the legumes, just, you know, probably within the margin of error. The grass seemed to be less. And you can see the grass went down at every sampling point. And that makes sense because this is Italian ryegrass, and which we don't think is a host for verticillium. And the verticillium just seemed to go down a bit over time and went from about, you know, 9,500 or 9,000 to under 4,000 in 2 years, 2 growing seasons. So that's quite interesting. The red clover and the alfalfa also went down, but then recovered a bit. So it seems like they go down a bit over winter, but they seem to respond a bit to the legume that is growing there. So they can be hosted in that legume crop. I'm not confident that these numbers are very much different than each other in reality, because the standard error on this verticillium testing can be quite high. So I'm not sure that these numbers in reality are a whole lot different than each other, but it does look like there's a bit of a trend for the grass to be going down. Now in the other field, you'll see that the grass, the verticillium in the grass went up. The proviso I will make here is that this was where that festolium treatment that I talked about, where the festolium didn't grow tremendous, and there was quite a bit of volunteer clover that grew in that treatment. So I think that may be contributing to why some of this verticillium went up. But again, the alfalfa is a little bit higher than the clover and the white clover. It is higher by about, 2,500 or so. Again, I'm not really sure in reality how much different that is. We will have to see. We will sample this again in May and see what the difference is, if we see much of a difference. The alfalfa looks maybe a little bit higher than the other two, but they're all still quite high. On the nematode side, though, we see quite a bit more difference. So this field, actually I get the places mixed up here. So this is actually the Huntley field, not the Alma field. So it starts off over 5,000, and you can see the red clover, it just goes up and up and up. And now we're up almost to 25,000. Compared to the alfalfa and the white clover, which are very similar in that they kind of stayed the same, went down a little bit, and then went up a bit last season, before the end of the season. I expect those will go down again over winter a bit, but they are half of the amount that we see in the red clover. And in the grass, again, they're kind of variable, a little bit lower than these two. As I said, this grass treatment's probably got a bit of clover in it, so that may be having an effect. This is actually the Alma field, not the Huntley field. Again, we see the clover goes way up. It then went way down, and then it went way up again. So it seems to me that red clover has a great ability to multiply nematodes. The alfalfa in this field basically stays almost steady the whole way through. It started off low and it's staying low. The white clover seems to be somewhat intermediary. It doesn't go as high as the red clover. It does, goes up a bit, but then stays fairly steady. And then the grass does jump and then comes back down a bit. So what are my take-home messages here? From our first 2 years of trial data here on the soil test, it appears that red clover multiplies nematodes faster than the other legumes. The root lesion nematodes in alfalfa, Timothy, were very low in one field, moderate in the other, but still lower than the red clover. And it does appear that grasses can still multiply the root lesion nematodes. The ryegrass and the fistolium still look like they had an ability to multiply root lesion nematodes. In the verticillium, it was a little harder to see. In the field where we had Italian ryegrass with very little legume volunteer in it, the verticillium tended to go down. Where we had some volunteer clover, it went up. But I can't really say we saw a big trend up or down on the legumes. On vert, it more or less seemed to stay more or less within a similar range. And we'll have to see what the effect is on the potato crop next year. So these will be tested in May. The soil testing will be done in May, and then it'll be in potatoes. We also are doing a biofumigation trial. So we set up 3 fields with Genesis Crop Systems with Steve Watts last year, 2 in West Prince and 1 in Central Prince, where we were looking at this biofumigant mustard mix of Caliente, Rojo mustard, and arugula. You can see here where there's sort of purple flowers in amongst the green ones, or purple leaves amongst the green leaves. The purple is the arugula. That we then compared that at 3 fields with Centennial brown mustard, sorghum Sudan grass, and the check of annual ryegrass. And then you can see we have a pretty decent fertilizer program on the mustard because you need that much nitrogen and sulfur to make the glucosinolates that are in the to maximize your biomass and maximize your glucosinolates in the mustard, which creates the gas for the biofumigation. They were planted in, as I said, in sort of late May, early June, and then incorporated in July for the most part, about 50 to 60 days after planting. We were actually surprised in some fields. We had to incorporate them even faster than we thought because they grew really quickly. And you have to make sure you incorporate them before they start making seeds, because once they start making seeds, they start moving the glucosinolates to the seeds. So you want to incorporate them while they're at peak biomass, and that glucosinolates is distributed throughout the plant biomass. Then about 2 weeks after the mustard was incorporated, we planted Sudan sorghum, where we incorporated the mustard, and that was left to winter kill. And then before anything was planted, we did all this soil testing, and we will do follow-up soil testing before the potatoes are planted this year. So this is just a brief little video, shows them going through and flail chopping the mustard. You can see it's actually quite high and quite dense, and it's gone through with the flail chopper. And then going behind is a lemkin, and that lemkin is incorporating right behind the chopper. So the aim was to try and incorporate and roll this residue into the ground directly behind to prevent any loss of off-gassing. We also had 2 more fields with the same mix that was part of Living Labs. So we had a total of 5 fields that we have a Caliente mix compared with a ryegrass check. And these are all going into potatoes next year. And we have plans to set up at least 5 more fields on this plan this spring that will be in potatoes next year. And then finally on mustard, we have a project again with Steve and Genesis and Ecota on looking at both the agronomic and economic values of Centennial mustard as a harvested crop versus a green manure crop. So there's a lot of people growing mustard for wireworm control. We understand from the good work being done by Dr. Nyiraneza that the majority of that impact on lowering wireworm is through it just being grown and present in the field, through its effect through the roots. And so could we look at, can we compare the difference of incorporating or mowing Centennial Brown Mustard versus letting it grow out and harvest it? And if there's not much difference in wireworm damage and there's not much difference in yield, but you can make money growing and combining and selling mustard, then we need to look at that. So again, we've got this at 3 sites. 2 sites have these all the sites have these treatments, and in 2 more sites, we also have sorghum Sudan grass alongside these as well. So we'll have more data on that when it's in potatoes this year. And I think Steve and Akota are looking for some more growers that are be interested in trying this trial in this year. So if you're interested in this, you're already a mustard grower and you'd like to maybe look at this, give me a call or give Steve a call, and we can probably make that work. So this is my thank you slide for the Living Labs and the main AIM part of the program. So thank you to our collaborators through Living Labs, particularly Agriculture and Agri-Food Canada, East Prince Agri Environment Association for they're the external lead on the program, on the project, and then Kensington North and Surrey Watershed Groups. They've been fantastic in terms of working with us on the sampling, and of course our AIM funding partners.